Hello everyone, this is Dr. Kingston, and in this video we'll be talking about the ventricular system and cerebrospinal fluid. In this video, we are going to take a look at what exactly cerebrospinal fluid is. We're going to talk about its function and see where it's located, and then we are going to describe the ventricular system that it flows through and discuss the role of arachnoid V-link granulations in maintaining flow of cerebrospinal fluid. So let's dig right in. What in the world is cerebrospinal fluid? Well, it is an ultrafiltrate of blood, which, without getting into too much or, any physiology, means that it is an already existing component of blood that has had everything else carefully removed. So that filtration process is accomplished by a vascular network called the choroid plexus, and that is going to be found in it lining a number of cavities or empty spaces in the brain called ventricles. Once it has been filtered out of the bloodstream, cerebrospinal fluid is secreted by the choroid plexus into those ventricles, and from there it flows into the subarachnoid space, which you will call fully surrounds the brain and spinal cord. So it makes sort of a water balloon-like cushion surrounding your central nervous system. In addition to that protection provided by the cushion, cerebrospinal fluid also provides nourishment for the central nervous system, and it provides waste removal. So it's multifunctional. To get to the arachnoid space, however, cerebrospinal fluid first has to flow through these interconnected ventricles. Now, I want to make clear here that the blue spaces that you're looking at on the slide here, they're not solid structures. These are empty spaces. They're just colored blue here so you can see them, okay? I also want to make clear that the choroid plexus that produces cerebrospinal fluid extends into all four of these. So not all cerebrospinal fluid is made in the lateral ventricles. There's some made in the lateral ventricles, some in the third, some in the fourth, okay? With that said, though, we are going to start working our way through the cerebrospinal pathway, starting at its most extreme end, which is going to be these lateral ventricles. So these are these C-shaped ventricles. They are very large, and they extend into the deep parts of the cerebral hemispheres. Okay? You may see these referred to as the first and second ventricles, but because that implies, one flows into another, and that's not the case, we call them both lateral ventricles. These are both connected to the third ventricle, so here's the third ventricle, by a little hole called an interventricular foramen. So cerebrospinal fluid produced here in the lateral ventricle goes through that interventricular foramen to get to the third ventricle. The third ventricle is then connected down to the fourth ventricle by a structure called the cerebral aqueduct. Okay, so here's the cerebral aqueduct, and that brings us down to the fourth ventricle here. The fourth ventricle sits between the cerebellum and the brainstem, and it is contiguous at its most inferior aspect here with the central canal of the spinal cord. All right, so that is a small cavity within the spinal cord that is filled with cerebrospinal fluid here. The fourth ventricle also provides some pathways out of these ventricles to the subarachnoid space, and it has three small apertures to do this. There are two laterally, two, and one medially that allow cerebrospinal fluid to enter into the subarachnoid space. All right, here's a quick view of what these ventricles look like on a real brain. So the lateral ventricles will be tucked up this way in the cerebral hemispheres. The third ventricle is right here, and I want you to know that this is just a very shallow depression. It sits between the right and left diencephalon, and it is a very, very thin little cavity. All right, the cerebral aqueduct travels down here, And then here's the fourth ventricle, a little bit easier to see here between the cerebellum and the brainstem. All right, so these really are just empty spaces, and in a living person, they will fill with cerebrospinal fluid, and that gives the brain not only a nice watery cushion along the outside, but also helps to support its shape from the inside as well. All right, so let's take a full walk through here. I'm going to use just a text map here to give you guys an example of a good way to take some of this information and rearrange it while you're studying again. All right, so we're starting in the lateral ventricles. And from the lateral ventricles, we're going to travel through the interventricular foramina. That is going to take us into the third ventricle, through which we will pass through the cerebral aqueduct, which will bring us into the fourth ventricle. And from the fourth ventricle, we have two options. The first is that we continue down to the extreme end of the fourth ventricle and continue into the central canal of the spinal cord. Or we can pass out through one of those apertures. So there was the one median aperture in the middle and two lateral apertures out to the side. This brings us through little holes called subarachnoid cisterns, and that carries us out into the subarachnoid space. It's quite a journey. Yeah. All right, so CSF is great, but it does not last forever. So you want to remember it's providing nutrition and removing waste from the central nervous system as well as cushioning it. So at some point, it's going to need to be recycled. So that choroid plexus that we just talked about is always switched on. It's always producing cerebrospinal fluid. So now, because of that, we need a way to push old cerebrospinal fluid out of the subarachnoid space to make room for the new stuff. And the way this happens is simply by forcing it back into the circulatory system that it came from through the dural venous sinuses. So the dural venous sinuses, as you can see here, sit very close to the subarachnoid space. You've just got this meningeal layer of dura and the arachnoid mater sitting between them. So what happens is that some of the arachnoid mater takes it upon itself to fold it. It's a really tight little tufts, and these are going to poke their way up and into the dural sinus. These are called arachnoid granulations, and as you can see in the illustration here, if you've got cerebrospinal fluid flowing around through the subarachnoid space, it can travel right through those granulations and back into the dural venous sinuses. Okay, so this is just a nice little pathway for cerebrospinal fluid to make it to its recycling date. These little granulations can form anywhere, but they're primarily found protruding into the superior sagittal sinus. So you can see a number of them in this picture here. All of these little dark granulations are arachnoid granulations poking up at the dura mater. In areas where many arachnoid granulations are present like this, the sinuses tend to dilate or become wider. They expand into what are called lacunae, which literally just means a space. So I guess that makes a lacuna within a sinus a space within a space. Either way, these are areas where the dural sinuses expand laterally to accommodate these larger numbers of arachnoid granulations. And that is the end of the material. So here is your review question. A blockage of the left interventricular foramen would prevent flow of cerebrospinal fluid from the left lateral ventricle to which other space? All right, the correct answer here is D, the third ventricle. Both of those lateral ventricles will flow into the third ventricle, and they each have their own little interventricular foramen that they use to pass cerebrospinal fluid down. All right, that is everything. Thank you very much for joining me.